Hi everyone, my name is Elizabeth Osterland, and the following presentation is a brief intro into endoplasmic reticulum-associated degradation in budding yeast. If anything is unclear to you, and you would like to read more on this topic, I've attached a review article by Way and Davis, 2010, in the reference section for this clip. Keep in mind that this review was written after the article I've analyzed by Sato and colleagues. So in this movie, I will basically be going over what is currently known about ERAD in yeast and what my article contributed to this knowledge. As you know from our introduction page, ERAD is an essential process for getting rid of aberrant proteins that would otherwise accumulate within the endoplasmic reticulum. In yeast, there are 3 separate ERAD pathways to handle proteins from different locations within the cell. First, there are proteins that are located within the ER membrane and have an exposed cytosolic domain where the misfold has occurred. These proteins follow the ERAD-C pathway. Secondly, there are proteins that are either located within the membrane and have a luminal domain where the misfold has occurred, or are completely solvated in the lumen and are misfolded, and these proteins follow the ERAD-L pathway. Thirdly, there are proteins that have a misfold within the ER transmembrane domain, and these proteins follow the ERAD-M pathway. Please do not confuse these 3 pathways to be involved in the mammalian systems, as is described by Michelle, Mike, and Maggie in their pages. In mammals, there are separate pathways like this, but since there are so many more proteins in mammals to be processed by ERAD, it is thought that many more pathways have evolved, making it harder to distinguish one from the other. There are 2 complexes located on the ER membrane that are involved in the ERAD pathways. First, the HRD1 complex is involved with the ERAD-L pathway and the ERAD-M pathway. On the other hand, the DOA10 complex is involved in the ERAD-C pathway. This is the ERAD-C pathway. For the membrane proteins that are misfolded in the cytosolic domain, indicated by the black X, chaperones within the cytosol, such as heat shock proteins HSP42, HSP26, and HSP70, and co-chaperones YDJ1P and HLJ1P are ERAD-C substrate specific and stabilize the cytosolic misfold for transport to the DOA10 ubiquitin ligase complex, which recognizes the misfold and ubiquitinates the protein. Here you can see the oligomer of ubiquitin that was added to the protein and is circled in red. From here, the protein is retrotranslocated, or in other words, it is pulled out of the membrane by the CDC48 ATPase complex that you can see in blue. Once the protein is out of the membrane and free to move, the ubiquitin group targets the protein to the proteasome, where it is degraded into amino acids that will be recycled in protein translation. So that's the ERAD-C pathway. My article of focus does not deal with the ERAD-C, as it does not involve the HRD1P ubiquitin ligase complex that they studied, and as a result, it will not be discussed any further. Just be aware that it does exist in yeast. Next, we'll talk about the ERAD-L pathway. When a misfold is located within the lumen, chaperones CAR2 and YOS9 recognize its glycan moiety, circled in red, and try to assist proper folding. If this does not occur, the HRD3P ubiquitin ligase of the HRD1 complex recognizes the fold and directs it to the putative retrotranslocan Sec61. The luminal protein is then threaded through the Sec61 retrotranslocan out into the cytosol, where it is ubiquitinated by HRD1P. indicated in the pink, and UB7 is also involved. The whole process is driven by the CDC48 ATPase complex indicated in blue. Similar to what you saw before, once the protein is out of the membrane and free to move, the ubiquitin group targets the protein to the proteasome, where it is degraded into amino acids that will be recycled in protein translation. The ERAD-L pathway was discussed in my article of choice because it involves the HRD1P complex. Some proteins that follow the ERAD-L pathway include CPY, KHN, and KWW. These proteins were used by Sato and colleagues to demonstrate the effect of HRD1P mutations on the ERAD-L pathway. Finally, the ERAD-M pathway is the main focus of this paper. When a misfold is located within the transmembrane domain, the ubiquitin ligase, HRD1P, shown in pink, recognizes the misfold via its transmembrane domain. This was shown in the paper that I chose to analyze by Sato and colleagues in 2009. Before their research, the ERAD-M pathway was largely a mystery. The authors knew that the ubiquitin ligases are usually involved in some way in the recognition of ERAD substrates. And they also knew that HRD1P ubiquitin ligase had to be located within the membrane, or membrane bound, for ERAD-M to occur. This led them to study HRD1P exclusively. Anyways, getting back to the ERAD-M pathway, after recognition, the misfolded protein is ubiquitinated by HRD1P and UBC7 and retrotranslocated out into the cytosol. Again, this process is driven by the CDC48 ATPase complex. Lastly, once the substrate is tagged with the ubiquitin oligomer and pulled out of the membrane, it can go by the normal degradation pathway, where it is targeted to the proteasome and the amino acids are recycled in protein translation. What's important to note here is that ERAD-M substrates used within the paper include HMG2P, PDR5, and Sec61-2. Now that we know the three pathways, let's take a look at the HRD1P structure, and specifically why the authors started to focus on HRD1P in the first place. See the structure in the top right corner and note that HRD1P spans the ER membrane multiple times and has a ring domain on the C-terminal domain and an N-terminal domain anchor into the membrane. The ring H2 domain transfers a ubiquitin, shown in green, from an E2 ubiquitin ligase to ERAD substrates. The authors wondered if the ERAD-M substrates were recognized by the HRD1P because it too was located within the lipid bilayer and also had many unusual hydrophilic residues located within the transmembrane domain, which you can see in the sequence below. They postulated that these residues would be involved in the interaction with exposed hydrophilic residues on misfolded proteins within the hydrophobic membrane. In the sequence, you can see the 6 membrane-spanning regions are underlined and highly conserved residues are shown in bold. To test their hypothesis, over 70 individual point mutations were introduced in the transmembrane domain of HRD1P. And each was changed to an alanine, and the effect on ERAD degradation was measured. The residues in blue were the ones they focused on the most in the article. So that's the end of the introduction, and now with this basic understanding about how ERAD works in yeast, you should be able to understand the figure analysis in the following presentations.